Hello students, so this is a plus 2 chemistry class and we were doing the chapter Biomolecules, and today in this we will start with the denaturation of proteins. Like in the last video we did that proteins is a polymer of amino acids in which large number of amino acids are linked together by a peptide polypeptide chain or polypeptide linkage, by which their mass is heavy mass more than 10,000 unit mass is present. Now, denaturation of protein means it is the loss of biological activity of proteins. When a protein in its native form is subjected to a physical change, means change in temperature, or a chemical change like change in a pH. Either it's in a physical change when we change the temperature, or it is in the chemical change when we change its pH, is known as a denaturation of a protein. When biological activity of protein is lost when it undergoes a physical change when we change the temperature, or it undergoes a chemical change like change in a pH. For example, like coagulation of a egg white on boiling. When we boil the egg, on coagulation of egg white part is known as a denaturation of a protein. Same way, curdling of milk. When milk changes into curd is also known as a denaturation of a protein. In this there is a change in the temperature. Temperature is changed when we boil the egg, and that white part, albumin part is coagulate, is known as a denaturation of proteins. Next are the nucleosides. Nucleosides means are those in which it's the combination of base and sugar. That sugar is present and one base is attached is known as a nucleoside. Two types are there, nucleoside and nucleotides. In nucleotide there is a base, sugar, and one phosphate group is attached, is known as a nucleotide, and this nucleotide is joined by a phosphodiester linkage, which helps in the formation of nucleoacids. So nucleoacids or polynucleotides. Nucleotides means in which a base, sugar, and phosphate unit is present. So what is nucleic acid or a polynucleotides? It is a long chain polymers of a nucleotides in which nucleotides are joined by a phosphodiester linkage between 5th and 3rd carbon atom of a pentose sugar. 5 carbon atoms are present. And these nucleic acids are of mainly two types, DNA and RNA. DNA full form is deoxyribonucleic acid and RNA is ribonucleic acid. Now in DNA, as name it indicates, it's the double strand helix structure in which two strands are coiled spirally in opposite direction, and in this sugar is present is D2-deoxyribose. And in case of RNA has a single stranded helix structure, sugar present is D-ribose, and it helps in the protein synthesis. Like it's the double stranded structure. Double stranded structure is of a DNA. This one is for a DNA. Double strands are present, helix structure of double strands, in which this is the helix of a sugar phosphate, and these lines, these indicates the base pair. What are these base pairs? In case of a DNA, these base pairs are, first one is thymine plus adenine and cytosine and guanine. These two are the base pairs which are present in the double strand structure of a DNA. And RNA is a ribonucleic acid which has a single strand structure. Only one single strand is present in case of a RNA structure. Now there are a few differences in the DNA RNA. First, DNA it is stored and replicate the genetic information. RNA converts genetic information contained within DNA to a format used to build proteins. This is the function of a RNA. DNA, two strands are arranged in the double helix made up of subunits of nucleotides. Nucleotides mean when one is the sugar, base, and phosphate units. And in RNA only one strand is present, and DNA is larger in size, the helix structure is larger, and RNA is shorter and only one single strand is present. Sugar in case of a DNA is deoxyribose sugar, and in case of RNA is ribose sugar is present in the structure of a RNA. So this is all about the that nucleoacids. Nucleoacids means long chain of a polymers, nucleotides, in which they are joined together by a phosphodiester linkage between fifth and third carbon atoms. Two types, DNA and RNA. There is a difference in their structure and their function also, and their base pairs are also different. In the case of DNA, double strand structure is there in which the base pairs are present on the structure of a DNA. Thank you.